Okay, so our next speaker, last but not least, Dr. Teitell from the University of California, Los Angeles. And he'll be telling us about the impact of polynucleotide phosphorylase on the therapeutic potential of mitochondrial RNA import.
So I'd also like to thank Aubrey for the kind invitation to come and the SENS Foundation for allowing me to present our work. I'm going to present the work in the manner in which we discovered the things that I'm going to tell you about, so you can see the sort of train of progression and thought. I'll get to the conclusion, and then I'll have a few slides at the end to show actually that we're able to use this technique to rescue mitochondrial mutations in relevant settings. So we're going to talk about PNPASE. It's a protein in the context of RNA import into mitochondria. I'm very new to the idea of rejuvenative medicine and don't claim to have a position one way or the other related to mitochondrial DNA mutations and their cause or effect in aging. Would like to point out that last night, Bill Andrews yesterday gave a really nice talk related to Ron DePino's work on telomerase and showing how that generated issues related to aging. What he didn't mention, in fact, was that paper included connections directly to the mitochondria and mitochondrial dysfunction. And so it may be very difficult, actually, to separate very cleanly the different aspects in aging one from another and figure out which is cause and effect. What I'm going to focus on today is simply mitochondrial DNA damage and perhaps a way to overcome that. Parenthetically, just at the outset, if you have this damage and you repair it, say, with a genetic approach to repair the DNA, how can you be certain, because you haven't repaired the source of the damage in the first place, that this won't happen again and again and again after you've repaired the mitochondria? So you might want to think about an alternative strategy related to not repairing specifically the DNA, but more of a complementary approach. So my lab is really involved in cancer cell biology. I'm not a mitochondrial biologist in training, by training, but about 9 years ago, we had this project and we discovered what I'll show you in 2 slides briefly, and then switch directly to PNPASE. So we noticed about 10, 11, 12 years ago now that in human B-cell lymphomas of a particular type, there was a tremendous amount of an oncoprotein expressed called TCL1, and you can see that here in the northern blot. And that was interesting. Just having a lot of protein expression doesn't mean it's causative in the cancer, but we made transgenic animals and actually developed these mice developed B-cell malignancies that were very important for the field. And the question immediately became, well, how is this very small protein that's not enzymatic, how does that protein cause cancer? And there was a lot of work by our lab and by many other labs about finding interacting protein partners. And we simply did an IP mass spec analysis and found this protein called PNPASE that I will focus on. I'm really going to be done in discussing TCL1 cancer and its interaction with PNPASE because, frankly, we don't know exactly how that interaction would promote cancer or retard cancer in any way. So what are PNPASes? So PNPASes are an evolutionarily conserved class of catalytic enzymes. They have 2 catalytic domains, RNase pH domains, separated by an H domain, which is an alpha helical domain. And this is actually defining for the family members. They have RNA binding domains, KH and S1, on the carboxy terminus, and they have targeting signaling sequences on the 5' end. In plant cells, it's very interesting. There's 2 PNPASes, 2 genes. One is targeted to the chloroplasts and the other to the mitochondria. And in humans, there's only a mitochondrial targeting sequence. In the plant, PNPASes actually regulate the turnover of RNAs that encode for genes that regulate photosynthesis and the light-dark cycle. So it's a very important enzyme. What does PNPASE do? PNPASE is a 3' to 5' exoribonuclease and also an RNA polymerase. So it can actually, in its polymerase activity, add nucleotides to the 3' end of a protein, of an RNA, sorry. And it can also extract or subtract nucleotides from the 3' end of an RNA. PNPASE is found in worms, flies, and vertebrates, almost every species, but it's not found in fungus. And that's really interesting because it gives us an opportunity to use yeast as a genetic system to study overexpression. Its expression can be induced by type 1 interferons, and that comes back to a lot of the theme of this conference, that inflammation has a role in aging. And this is one of the proteins that is induced in an inflammatory response. As I said, there's homologs in chloroplasts. This is the reactions that it indicates. There's a polymerization reaction, and there's a degradation reaction that PNPASE can foster. And for RNAs in chloroplast, it's been shown that the polymerization reaction is actually required to add adenine dinucleotides or adenine nucleotides to the 3' end of an RNA to make it a target for degradation. So it makes it actually an instability sequence, as opposed to what you'd think about with polyadenylation from nuclear-encoded genes, making it a stability sequence so it can be translated into protein. What does PNPASE look like? All of our modeling and structures are based upon the crystal structure of several forms of PNPASE from the bacteria, but it pretty well holds. PNPASE, these 85 kilodalton proteins, exist as trimers, and the trimer is shown here, where the RNA binding domain is on one face of the molecule, and this thing forms sort of a planar donut with a central cavity. And that central cavity is actually the catalytic core. So RNA is bound here and fed into that core where it can be polymerized and degraded and so forth. It turns out that these donuts can stack one on top of another and form sort of a hexamer of the same monomer. And that gives you a very big channel in which you can do a lot of RNA processing. And no one knows which form exactly exists within cells. These are crystallized structure properties. Bacterial PNPASE can exist on its own, as you see here in the crystal structure and also in the modeling. And it can also exist as part of an exosome. The human exosome has unique properties and unique proteins, and you heard a little bit about that earlier today, but it does not contain PNPASE. So PNPASE in human cells exists on its own and not in the exosome. So where is PNPASE? Even though there was a sequence that looked like a canonical mitochondrial targeting sequence on the 5' end, it was never shown or proved to be a mitochondrial encoded protein. So we used a couple of methods. I'll just show you one here. What we did was we used hypotonic lysis of the outer membrane of the mitochondria to ask if PNPASE, if it was targeted to the mitochondria, would reside in the intermembrane space zone or within the matrix of the mitochondria. The way you can do that is you can make this hypotonic solution, treat the mitochondria with that, spin down the mitochondria and isolate the supernatant, which contains the IMS proteins, and the pellet, which contains the intermembrane, the matrix, and the outer membrane proteins coming down on the pellet. If you add protease and it's an intermembrane space protein, it'll be degraded. If it's a matrix-associated protein or if it spans the intermembrane, it'll be protected from that protease, and so you'll see it. This is the result. This is not so important, but this is the important guy. Apoptosis-inducing factor is an intermembrane space protein. As you can see, as we make the solution more hypotonic, it gets lysed, and you can see the same thing for PNPASE, whereas a heat shock protein 60, which is a matrix-associated protein, is protected from lysis. So this is a nice, simple way to show where a protein is localized within the very complex mitochondria that was beautifully described by Michael before. We used other methods as well, including protein import assays to localize where the protein was imported, and confocal microscopy with immunofluorescence microscopy to show also that this is an intermembrane space protein. That's a very unexpected localization for this protein. I mean, it has an RNA degrading activity, and yet it's localizing to a position where there's ostensibly no RNA. You know, the mitochondrial genome with its 13 genes is in here, and if it's making those genes into a polycistronic message that then gets processed into RNAs, you'd imagine that RNA degrading enzymes would be in here. So it took us about 2.5, 3 years to actually convince ourselves that this was right. If you knock down PNPASE with an RNAi approach, you can see the extent of knockdown. You actually can't eliminate PNPASE from human cells. It's lethal to the cells. They senesce and then they ultimately die. Same thing for mouse cells. But if you knock it down, what you can show is that the activity, the respiration activity, oxygen consumption for length complexes 1 and 3, 2 and 3, or 4, is about 40% of the wild type. So it really has an impact on respiration of the mitochondria. It also makes them these cells less sensitive to inhibitors of ATP synthesis. So PNPASE has a very big role in the regulation, somehow, of the mitochondria for energy production and oxygen utilization. very striking is if you grow these RNAi PNPASE cells in culture over time, it's very easy to appreciate that this control colony with a scrambled RNAi has the usual pink color of the media, whereas the RNA knockdown of PNPASE is very yellow and actually filled with lactate. So there's a big change in the metabolism of the cells, as you can appreciate here. You can see it's quantified here. The amount of ATP that's produced in PNPASE knockdown cells is about half or less that you see in wild-type cells. And because of that, that triggers the phosphorylation of AMP kinase family members, which is a regulator of energy stress inside cells. And that also will feed back into the nucleus and slow down the proliferation of cells. And you can see that here as an example by day 3 or day 6. The PNPASE knockdown cells are not growing very fast at all. In fact, they begin to senesce, and ultimately they'll die. So to this point, a model for the effect of PNPASE reduction, without telling you how it does what it does, is that you have the mitochondria with its electron transport chain and its ATP synthase, a mitochondrial charge, electromembrane potential. You knock down PNPASE, you reduce the amount of these complexes, and what happens is that, and I didn't show you this data, but this ATPase dissociates, and so ATP is not easily generated from it. The membrane potential drops. I also didn't show you that the mitochondrial network, which is usually a fused network in somatic cells, becomes fragmented. This leads to a reduction in energy charge in the cell, an increase in the use of glycolysis to maintain the cell's viability, which increases the amount of lactate that's present. This drop in energy charge causes the phosphorylation of AMP kinase and slows down the rate of proliferation, so the cells senesce and ultimately they'll die. So to this point, knocking down PNPASE causes a decrease in oxygen consumption, respiratory chain activity, ATP to AMP ratio, and increased lactate production and glycolysis, AMP kinase phosphorylation, and the slowing of the cell cycle. So how does PNPASE do this? And as I told you, I did not show you, but it controls both the function of mitochondria and the structure of mitochondria. This was a very hard and daunting question for us. The first thing we established as best we could with native gels was that PNPASE was in a multimeric complex, but it actually was in a homomultimeric complex. We tried just about every way under the sun to find other proteins that would interact with it, and we couldn't localize any other proteins by IP approaches with mass spectrometry. This size of the complex would be the trimer. Perhaps these larger smeared bands would be a multimer, like a stack of trimers or a hexamer. And there might be reasons I can get into why PNPASE doesn't easily co-purify with other proteins, but we were not able to do that. So we made a knockout mouse. The knockout mouse, we did it by flanking exon 2 with LOXP sites because we were very concerned, since we had very big trouble with the RNAi knockdown expression constructs, that the complete knockout would be embryonic lethal. So rather than just making a standard knockout, we made the potentially conditional knockout, but we first test whether or not the full knockout is embryonic lethal, and it is. You expect about 25% in the genetics of this cross right here, and we got 0% observed. So you can't knock it out of mice. What you can do is you can cross it to a promoter that drives the expression of Cre recombinase linked to albumin, and so you can knock it out of the liver. And we reasoned that we'd want to knock it out of the liver because the liver is a relatively highly regenerative tissue. And so if it was at all to be tolerated, perhaps the liver would tolerate the knockdown of PNPASE in most of its cells. No Cre recombinase is completely 100% efficient, and the liver could regenerate some of itself to rescue sort of the liver's function. So what you see with these liver knockdown mice, actually we call them HEP-KO mice for hepatocyte knockout, is you see a nadir, you see a trough in PNPASE expression, and then the trough relieves itself. The trough's around 5 or 6 weeks. So we do all of our experiments there. Then the liver regenerates, and you see plenty of PNPASE again. There's a real competitive advantage for having PNPASE expression. What does the liver knockout hepatocytes look like? By electron microscopy, they're usually smooth, stacked linear cristae, which indicates how their electron transport chain functions. Now gets replaced by these circular, irregularly shaped cristae that don't stack and they're disorganized. We see this only in the knockout. We don't see it in the wild type. And if you look at the respiration, like the knockdown in cells, you can see about a half defect for the amount that we can knock down PNPASE in these mice. So this is a physiologic effect, which is nice for us. But again, I haven't told you at all about how PNPASE does this. And the first clue we had into how PNPASE might be doing this is in both the knockdown or in the knockout for RNA expression and also protein expression. So on the left panel in the knockdown, what you can see is for all the genes that we checked, and we checked the others as well, the amount of RNA that's encoded within the 13 genes that code for proteins within the mitochondria is reduced in the PNPASE knockdown. It's a little harder to appreciate, but these gels are pretty standard. This is a gel that's actually showing you a smear of all the proteins, again, that are encoded within the RNA, within the mitochondria by a mitochondrial genome. And you can see that in the knockdown, it's really, we call it a knockout, but it's really a knockdown. In the liver cells, there's about half the amount of proteins present. So there's a generalized reduction in RNA and protein in the knockdown cells, or in the knockout mice. More interestingly, much more interestingly, is how the mitochondrial genome is transcribed and translated. So the mitochondrial genome is transcribed off of two main promoters, and it's transcribed as a polycistronic message. You don't see that in the nucleus. You only see that in the mitochondria in mammalian cells. And that polycistronic message has got to be processed into its individual RNAs for translation to occur. And what you can see here is we designed primers that span one of these processing sites, if you will, between the COX-1 and the COX-2 gene encoded within the mitochondrial DNA. And what you can see down here is when we use the COX-1 forward and reverse primers, you get equal amounts of this guy, just the internal control, if you will, in the wild type and the knockout. But if you now look across the junction, what you see is the wild type's completely processed and the knockout isn't. So there's a defect in processing. You can show that both by qPCR, and people in this field hate qPCR because it's so sensitive, so you have to show things with western blots and northerns and so forth. And so you can show exactly the same thing with a northern, where these larger precursor unprocessed forms stack up in the knockout, but not so much in the wild type. That's really interesting, and that caused us to take a very big leap of faith. We have an RNA processing enzyme in the intermembrane space without a function known in the intermembrane space. And we have an RNA processing defect within the matrix. Could there be a connection? The connection we dreamed up was that there are some RNAs, RNase pRNA, mrpRNA, that are imported, known to be imported from many years of work, into the mitochondrial matrix to assist with DNA replication and with RNA transcription and translation and processing of polycistronic RNA messages. And we guessed, really, that PNPASE might affect this import. That's the best I can tell you. And so in the hepatocyte knockout, what you can see is that this RNase pRNA encoded in the nucleus is now reduced in its import activity. More interesting, and kind of a nutty experiment if you think about it, we have an RNA degrading enzyme. We did an experiment in cells where we overexpressed, tagged TIM23, which is an intermembrane space protein, as a control, or PNPASE, and then added RNase pRNA, COX-1, GAPDH, 12S ribosomal RNA, or a tRNA that's not imported normally, and did pull-down assays and asked, does any of these proteins, do any of these RNAs bind to PNPASE? And the only one that bound to PNPASE was RNase P, as shown here. This is coded inside the mitochondria. This is never imported into mitochondria. This isn't, and this isn't. It's very curious that this binds but is not degraded. It's a degrading enzyme. Similarly, if we take HEC293 cells, add these tagged constructs, and then feed them radiolabeled substrates, a random RNA, RNase pRNA, or mrpRNA, and ask who binds, it's the PNPASE lanes for RNase P and mrp that bind. Indicating that, and this must be at the core of the function of this thing, that there's an RNase activity, a processing activity, that somehow these guys that get imported don't activate, and instead bind the PNPASE, and somehow that mediates import. And we're working very, very hard on that. But it's an odd thing to have, quote, a receptor that's an RNA degrading enzyme, not degrading RNA, selectively. As I mentioned before, yeast lack PNPASE, so we can do gain-of-function studies inside the yeast. Just to make sure that this would be a reasonable thing to do, we simply looked at the yeast to know whether or not adding PNPASE would affect any of the typical physiology of the yeast. You can see how much PNPASE we can add. It's a little hard to see, but the vector alone or the PNPASE addition doesn't seem to affect the mitochondrial morphology in yeast. It doesn't affect the rate of growth of yeast, and it doesn't affect the survival of the yeast as well. I didn't mention this earlier, but the import machinery for proteins in mitochondria is conserved across species, and we guessed that the RNA import machinery would be conserved even though yeast lacks PNPASE. So if it works, it means that PNPASE can augment or mate with the endogenous usual RNA import machinery of yeast. The way we did these experiments was to add a radiolabeled precursor RNA to isolated mitochondria. ATP and intact membrane potential is required, so the yeast mitochondria have to be functional. And then we can add protease to remove all the things that are imported. What you can see is that in isolated yeast mitochondria with PNPASE expressed, the import is pretty heavily augmented, which is nice. We don't expect the import to be absent in the vector alone because yeast can function fine without PNPASE. GAP-DH RNA, which is not imported normally in mammalian cells, is not imported if you add PNPASE as well. The imported RNA is protected from degradation. Protease assay right here, this lane right here. Here's RNase pRNA added. It's protected from degradation, which is important. It says that the RNAs are being imported into the matrix and not stuck somewhere in the IMS where the PNPASE is localized. If you knock down the membrane potential, you lose all the import activity, so you require functional mitochondria. And if you have PNPASE present in the yeast, or you don't, you augment the import when you add 1 time or 2 times the amount of RNA for the import assays. And this is equal loading of protein, showing we're having an equal loading of the mitochondria that are present as well. And it's dose responsive. PNPASE has this polymerization and degradation function, and in work from Gaddy-Schuster's lab, not from our lab, he did biochemical analysis to find out whether or not where the residues for the polymerase and degradation activity resided in the human PNPASE molecule. And he could show that if you make a mutant at 484 where you change a serine to alanine, you lose polymerase activity and degradation activity. We took advantage of that to make mutants in yeast and asked, could RNase pRNA be imported? And in the mutant that lacks degrading and polymerase activity, import still occurs. We had to verify that this was a degrading defective mutant, and so we showed that here, where you get a massive reduction in the ladder of degradation from the wild-type PNPASE. The imported RNA is just as stable with the mutant as it is with the wild-type, so it's not an issue of stability or accumulation of RNA related to stability. It's true import from the PNPASE molecule. So what's special about these RNAs is don't trigger the degradation, but instead allow an import to occur. We asked that question, and we thought it would be encoded in the RNAs. So we did deletion mutants. And what you can see here is that this region right here, which is deleted, is the key region because it doesn't allow PNPASE to import RNA if it's absent. All the other regions that you can see over here do. And the same sort of thing, we can do a pull-down assay with that region. These small regions here generate RNA hairpins that are found in RNase-P and MRP, which are imported as RNAs. And they can be used, actually, to transfer the import activity. I didn't show that, but here's GAP-DH right here. And with the control sequence, there's no import. With the MRP sequence, there's import. With the RP sequence, there's import. So we can easily do the import assays. We can do all this work in vivo in humans and actually get rescue or loss with the knockout of import of the RNA. And the HEP knockout cells lack RNA import, whereas the wild type have the import. So this was a cartoon, and I know I'm out of time, but I'll go through this very fast. RNA is imported. It gets involved in mitochondrial DNA replication or processing of RNA to allow the electron transport chain to function and the cells to grow. Finally, I have to show you this. The reason for being here really is we have a paper that's now under review, so I can't give you many of the details, but will this work as a therapeutic? And the answer is unequivocally yes. Milos and Murph are 2-point mutations in mitochondrial tRNAs that don't allow the translation of the mitochondrial genome. We use cybrid lines for rescue experiments in this regard. If you take the little stem loop sequence I showed you before and add it to a tRNA that encodes for lysine to rescue the Murph phenotype, we can show in isolated mitochondria in wild type or knockout that it gets imported into the wild type and not into the knockout. Same thing over here for the Milos phenotype. And if you do an inorganol S35 incorporation assay, you can see that now it rescues the ability to translate the mitochondrial genome. More interestingly, more importantly, is you can do the exact same thing with a mitochondrial encoded gene, COX2, which is 683 nucleotides. So it's a very large RNA to actually try and import. And because time is short, I can just show you down here that appending that stem loop sequence allows the import to occur. 3 elements are actually acquired for import: nuclear stabilization, mitochondrial localization sequence, and mitochondrial import sequence. You have to have all 3 of those. We have those in these constructs. I can't go into the details. More importantly is the protein is actually expressed within the mitochondria, and you can do that by Western blot. It rescues expression, as I show you here, by mitochondrial protein translation. It rescues specific protein expression, as you can see here, by Western blot for mitochondrial encoded genes. And most significantly, it rescues the function of the mitochondria. So oxygen consumption is increased by about 70%. So this is the final slide. In summary, PNPASE binds the T-cell oncoprotein. It regulates the import of small nuclear-encoded RNAs. It impacts mitochondrial RNA processing. Its degradation is separable from its import function. It regulates cellular energy charge and proliferation. And it really looks promising as a therapeutic pathway to nullify mitochondrial mutations with age and also in diseases, of course. We've tested in diseases so far. And the method really resembles evolution in shipping things out of the mitochondria, as was pointed out by Michael earlier, and then bringing them back in. We're excited because we don't have to worry about the ongoing mutations in the mitochondrial DNA because we can overcome that by simply importing the wild-type RNA. Guys who did the work are here. All the work has been revalidated independently by Nina and Yvonne in CERN Strasbourg. And we have a collaboration that I haven't talked about at all with Christian Kubisch in Germany, where he's isolated a family that has a mutation, a point mutation in the PNPASE catalytic domain. It limits RNA import, and it causes a neurodegenerative disease, and we're trying to get that published now, too. So thank you very much.
We have time for a couple of questions.
Wonderful talk. I have a crazy idea. I don't want to compete with Aubrey, but I have a crazy idea, and I want you to tell me why it's wrong. RNA, as I'm sure you're aware, but many in the audience might not be, has an intricate set of controls for misfolding. And they're equivalent to HSPs in effect. Okay. Where would misfolding be a serious problem? Well, one place is in the middle, in the midst of mitochondrial import. You get a tangle that would block the access for import. And maybe dwelling in that transit space, having something that is both an exoribonuclease and an RNA polymerase is to do exactly that, to repair or at least get rid of the tangle so that you unblock the pore.
Not a bad idea. I didn't tell you what the pores were because I don't know yet, but we have some ideas. Also, proteins are imported by unfolding, by denaturation, and we doubt that the tRNAs will be imported by that mechanism, so there has to be a nicely large-sized pore for these things. And the secondary structure for something like COX-2 is certain to be extraordinary. We thought about a proofreading or an editing function for PNPASE in that space, similar to what you're saying, which makes a great deal of sense. The yeast data kind of argues against it. If it's just degradation and not active import, we'd expect a reduction in the import and we see an augmentation. But in terms of editing RNAs that are being imported and making them better, if you will, as import substrates, sure, that's not unreasonable at all.
I mean, maybe yeast just don't need it because they're dividing all the time anyway, so they can kind of dilute away any mitochondrial problem.
Perhaps, yeah. And the other nice thing, too, I couldn't talk about it again because of time, but you really only need about 20% fix to get a full repair in the phenotypes. That's pretty nice.
That was really beautiful. I'm actually really jealous. Maybe this is a naive question, as I'm also new to the field, but it doesn't surprise me if you're on the knockdown that you're losing respiration, and the activity of these, but the really dramatic structural morphological change that you saw. Do you think that the impact just on those 13 proteins is sufficient to explain that? Or do you think there might be something else?
Yeah, no, it's a great question. There's huge chicken and egg issues with all things mitochondria, as you're almost certainly aware, Michael. If you cripple the membrane potential, you're going to get fragmentation of the network. This thing definitely cripples the membrane potential when you knock it down significantly. The thing that kind of saves us to give us this as the primary mode of operation is the time course. So if you knock down PNPASE, the nadir, the trough in a knockdown, not in a knockout mouse, but in a knockdown cell, is about 7 days. If you do experiments before then, you don't see any of the physiology, so no fragmentation, no loss of membrane potential, no loss of respiration. But you do see import defects. And in fact, Nina and Yvonne pointed that out to us. So it's the first thing that happens is the import is affected, probably leading to the inability to continue to make the mitochondrial respiration chains. You still run the guys that are there until they're unstable, get switched out, and then you lose what you need. So we think that those effects are secondary and the primary effect is important.
Last question.
Do you see any changes in PNPASE expression in aging populations, and is it associated with any diseases as yet?
So I can answer the second one easily. The first one, we haven't looked in aging populations. It's expressed in all cells and all tissues of the body, in the mouse and the human. So that it's required actually for mammalian cells, not required for yeast, obviously. And a collaborator of ours, Christian Kubisch at Ulm University in Germany, identified 2 families that have point mutations in PNPASE, and they cause a neurodegenerative disease. We did the import assays for them, and it actually reduces the importing RNAs. It can't be you're not going to get a null with this guy, I think, because you're not going to get a person. Great.
Thank you very much, all.